Hello and welcome to IB Biology Notes with Dr. Kimmo. This is A2.1 Origins of Cells. This subchapter is additional higher level, and it is about the origin of life and the origin of cells. What we know, what we think we know, and what we don't know. Prebiotic Earth. Prebiotic means before life. The solar system formed from a spinning disk of gas and dust about 4.5 billion years ago. Earth's first atmosphere was very different from the one we know today. There was a lot of methane, a lot of carbon dioxide, and almost no oxygen. Methane and carbon dioxide are greenhouse gases, so the Earth was very hot, although the sun did not shine as brightly as it does today, but about 20% less. Low oxygen levels meant that there was no ozone layer. The ozone blocks UV radiation from the sun, and without an ozone layer, the radiation levels are very high. There was possibly more lightning and probably more volcanic activity. So the Earth seems to have been a very dynamic, hot, energetic, and violent place. As a result of these special conditions, certain chemical reactions may have occurred and carbon compounds may have formed. For example, aldehydes, carboxylic acids, amino acids, and nitrogenous bases. These carbon compounds became the building blocks of life. As life evolved, conditions changed. Photosynthesis led to high oxygen levels and low carbon dioxide levels. The high oxygen levels led to the formation of a protective ozone layer, which led to less UV light reaching the Earth. Less UV meant that life could move from the ocean onto land. The low carbon dioxide levels meant a reduced greenhouse effect, and the Earth became more similar to what we know today. Life could not evolve again on today's Earth. Conditions are so much different from what they used to be, and if life somehow would evolve again, if some kind of proto-cells would emerge, they would be outcompeted by other organisms that are already here. Life on today's Earth consists of cells. Cell theory has three main points. 1. All organisms are composed of one or more cells. The tree that you see here obviously has many cells. The bacterium has only one. 2. The cell is the fundamental unit of organization, structure, and function of organisms. Of course, we can look inside a cell and we'll find organelles and a lot of things, but none of that is viable. The mitochondrion here that you see, it cannot survive on its own. So the cell is the smallest unit of self-sustaining life. 3. All cells derive from pre-existing cells. Every single cell has derived from another cell, and that cell derived from another cell, and all the way back to the first cell. Today, cell theory is widely accepted, but it was long believed that living organisms could emerge from non-living matter and that it happened all the time. For example, flies would emerge from rotten meat. This was called spontaneous generation, and it was kind of logical for people to think that it kept happening because you cannot see fly eggs and bacteria, for example, with the naked eye. Then came Louis Pasteur and showed that spontaneous generation does not happen. He conducted some famous experiments with swan-necked flasks. He heated broth inside swan-necked flasks in order to kill any living organism that was present. And when he left the flask standing, there was no bacterial growth. He made the necks very long and curved so that no living organism would be able to enter the flask. And, well, he had broth without contamination, no growth. However, when he broke the neck of the flasks, organisms grew in the flask after some time. He had bacterial growth. And these bacteria obviously came from the air in the room. And they were able to enter when the neck was broken. They were not able to enter when the neck was not broken. Alright, so Pasteur showed that spontaneous generation does not happen, except it did happen once. How? How did the first cells emerge? As you may have guessed, there is no complete answer yet. But observations and experiments in fields like molecular biology, genetics, chemistry, geology, and astronomy provide clues. Cells are made of molecular building blocks, mainly carbon compounds. In the 1920s, Alexander Oparin and J.B.S. Haldane hypothesized that the conditions on the early Earth allowed a variety of carbon compounds to form spontaneously and create a primordial soup. The idea that life has chemical origins is called abiogenesis. Could carbon compounds really form in a primordial soup? Stanley Miller and Harold Urey wanted to find out, so in 1953 they attempted to simulate the conditions of the early Earth. This is what their experimental setup looked like. At the bottom right you can see the ocean, water in a flask. They heated the water and the water vapor went up through the tube all the way to the atmosphere. In the atmosphere, besides water vapor, there was methane, ammonia, and hydrogen gas. And this was based on what they knew and believed that the atmosphere was like when life emerged. They also added electrical sparks to simulate lightning. Then the atmosphere was cooled, and the cooled water went back to the ocean, and the whole circle started over many times, over and over again. To their surprise, Miller and Urey could conclude that their experiment produced many carbon compounds, including more than 20 different amino acids, and they are of course the building blocks of proteins. This was quite revolutionary at the time. How do we go from carbon compounds to protocells, primitive cells? Well, there are 4 basic requirements for a cell. 1. A boundary between inside and outside. Today we have the cell membrane. It's very important. It determines what goes in and what goes out, and it maintains specific conditions within the cell. 2. Control over chemical reactions. Today we have enzymes which determine which chemical reactions happen. 3. Self-assembly from available materials. That would mainly be building polymers out of monomers. For example, building RNA strands from nucleotides. 4. Self-replication of molecules. Today we have DNA. DNA replicates itself in modern cells. Let's consider the first requirement at the top right, a boundary between inside and outside. Cell membranes consist of phospholipids, and phospholipids are amphipathic. It means that they have a hydrophilic part and a hydrophobic part. Because of the shape of the phospholipid molecule, we call the hydrophilic part the head and the hydrophobic part the tails. The hydrophilic head has an affinity for water, and the hydrophobic tails are repelled by water. So in a polar substance such as water, the phospholipids tend to assemble into bilayers, where the hydrophobic tails are on the inside, protected from the water, and the hydrophilic heads are on the outside, in contact with water. If you have enough of these phospholipids, then the bilayers might form vesicles. And there you have it. This might be how the first boundaries formed, spontaneously. In modern organisms, DNA contains the hereditary information. The information is transcribed into RNA and then translated into protein. And this is called the central dogma of molecular biology. Now you need DNA to make protein, and you need protein to replicate DNA. Then you have RNA there as a middleman. So which came first? Well, the leading hypothesis is called the RNA world hypothesis, and let's have a look at that. Let's first consider requirements 2 and 3 at the top right: control over chemical reactions and self-assembly from available materials. In modern organisms, metabolic reactions, including the assembly of polymers, are catalyzed by proteins, more specifically enzymes. It turns out that RNA has similar properties, and we call such RNA molecules ribozymes. And I think you're familiar with ribosomes, rRNA. They catalyze the assembly of amino acids into proteins. RNA, therefore, might be the first catalyst. Let's now consider the fourth and final requirement at the top right: self-replication of molecules. In modern organisms, DNA contains the hereditary information. However, RNA has similar properties, and some viruses still use RNA as their genetic material. So RNA might also be the first genetic material. RNA could have performed the job of today's DNA and protein, and that is called the RNA world hypothesis. Here's an overview of how life emerged and developed. First, a habitable world. The conditions needed to be right, for example, liquid water. Then prebiotic synthesis, so synthesis of molecules before there was life. Then polymers and vesicles formed. At some point after that, protocells, primitive cells, evolved, and one of these cells then started diverging and gave rise to the diversity of life. And this first cell, and that is the ancestor of all life today, is called Luca, the last universal common ancestor of all life. And the two main reasons that scientists conclude that life emerged only once and that all life is therefore related is that all organisms use the same genetic code, which is quite amazing, the same 4 letters. And that many other features are shared among all organisms, for example, ribosomes and some enzymes. Luca, the last universal common ancestor, is here at the root of this phylogenetic tree. It's a very simplified phylogenetic tree, showing the 3 domains: bacteria, archaea, and eukarya. And you can see from this tree that bacteria branched off first from the rest of the living organisms, and later the other branch divided into archaea and eukarya. On the right you can see a drawing made by Charles Darwin in 1837. Here the common ancestor of all life is marked with the number 1. We have discussed how life may have begun, but when did it happen? And how can we find out? You may have heard about radiometric dating. Rocks can be dated by comparing ratios between naturally occurring radioactive isotopes and their decay products. This method can provide the absolute age of a rock. The oldest uncontested evidence for life are rocks called stromatolites that formed 3.42 billion years ago. What you see here in the background are modern-day stromatolites in Australia. Stromatolites are formed by cyanobacterial mats. They consist of calcium carbonate formed by the cyanobacteria and of sediment. And as the cyanobacteria grow, they produce layer upon layer. Stromatolites can fossilize, and what you see here on the left is a stromatolite, a fossil stromatolite more than 3 billion years old. Let's have a quick look at how the dating works. For example, uranium-235 decays into lead-207. And the half-life of uranium is 704 million years. It means that after that time, half of the uranium will be gone and will have turned into lead. So after 704 million years ago, the ratio between uranium and lead is 50% to 50%. After twice the time, 1,408 million years ago, or 1.408 billion years ago, the ratio is 25% uranium, 75% lead. And after another 704 million years, the remaining amount of uranium has halved again. And what we have is 12.5% uranium versus 87.5% of lead, and so on. Another method is based on genetics. Mutations tend to occur at a standard rate, so as time goes on, there are more and more mutations. By comparing the base sequences of genes between species, it is possible to calculate approximately when they diverged. This technique is called the molecular clock, or the evolutionary clock. Data from several genes can be used in parallel, and the molecular clock can also be calibrated against fossil data. Imagine that a shared ancestor of two modern lineages lived 50 million years ago. Have a look at the sequence at the far left. After 25 million years, the two descendant lineages have diverged. Each has had a single base mutation, so that the lineages now differ by 2 bases. After 50 million years, the two descendant lineages have diverged further by another base mutation. And today, the lineages now differ by 4 bases. Molecular clock data suggests that Luca lived 3.5 to 3.8 billion years ago. Luca was probably not the first living organism. Older life forms have just gone extinct. The phylogenetic tree shows Fuca at the root of the tree, and that is short for first universal common ancestor. You can see from the tree that Fuca gave rise to Luca and to other ancient cells that we basically know nothing about. There may have been horizontal gene transfer between the groups. An example of horizontal gene transfer is what bacteria do when they share plasmids. All of this is based on genetics, but scientists also compare proteins. At the bottom left, you can see cytochrome C oxidase, which is involved in the electron transport chain, and it contains highly conserved proteins. These can also be compared between species and groups. Also, comparing ribosomes at the top right can reveal structures that are common to all species and were probably present in Luca. Here's an overview of some of the clues that can help us answer the question, when did life emerge? Stromatolites, 3.42 billion years ago. That's based on fossil data. That's the oldest uncontested evidence of life that has been found. And molecular clock data suggests that Luca lived 3.5 to 3.8 billion years ago. And this makes sense, of course, that Luca lived before the oldest evidence for life that has been found. The oldest rocks on Earth are 4.0 billion years ago. So it's very unlikely that we would find any clear fossils that are older than this. So that sort of sets the limits, not for the emergence of life, but for the fossil evidence that we can find. However, there are silicon crystals with a carbon isotope ratio that hints at life. These silicon crystals are older than 4 billion years, and this could be evidence of the oldest biogenic carbon. And scientists suggest that these silicon crystals may be the remains of older rock, which then melted 4 billion years ago. However, these silicon crystals, they were very durable, so they survived this melting. Also in silicon crystals, there might be the oldest signs of water. Some silicon crystals that are 4.3 billion years old have an oxygen isotope ratio that hints at water. Well, this suggests that at least it's possible that life was around back then or emerged a bit later. It doesn't tell us specifically anything about life itself. Then 4.5 billion years ago. That sets the definite limit for when life could have been around, could have emerged. Because that's when Earth and a planet called Theia collided. That's when the moon was formed. And this was such a catastrophic event that if any kind of life would somehow have been around before that, then probably it was wiped out. However, scientists don't think that there was life before this event anyway. Now, what points to this event, that this event happened, is similarities in the composition between Earth and the moon, and also traces of Theia inside Earth. And these traces were reported very recently. They had been suggested before, but there's a very recent paper reporting these traces of Theia inside Earth. Let's round up with this question. What was Luca like? A comparison of many species of archaea and bacteria have revealed many proteins that are very common in prokaryotes alive today. 355 protein clusters are present in at least 2 phyla in each domain. Each domain means both archaea and bacteria. They are not believed to have evolved separately many times. Instead, they were probably present in Luca. The selection of proteins indicates that Luca was thermophilic, anaerobic, and methanogenic, and that it lived in an environment rich in hydrogen gas, carbon dioxide, and iron. These clues point to a prokaryote in a hydrothermal vent setting on the ocean floor. That's all for now. See you next time.